but I will try to dig in a bit more in the examples of constructing a protein film on an electrode surface, how to interface proteins on this electrode surface, especially enzymes, and taking examples from my own experience, so I can probably give you some details of pitfalls and difficulties and problems that you can encounter while constructing that. So if we want to explore electrochemically redox proteins and redox enzymes, then we need to, the first hurdle that we need to tackle is the fact that these proteins, they have been, they have evolved to interact with other biomolecules and not with electrode surfaces. So you have to think about that you have to construct the connection, the interface between the protein and the electrode surface to try to mimic as much as possible the interactions that this protein has with other proteins, for example, with the redox partner. Nevertheless, we can see that proteins in vivo are actually embedded into a very crowded environment. They are surrounded by lots of proteins, they are surrounded by membranes and so on. So it's not so far-fetched to think that being immobilized on an electrode surface surrounded by other protein is such an alien environment, because in nature they're already quite close. They are already in a very crowded environment. The other aspect is that protein, especially enzyme, redox enzyme, they have evolved to protect their carrier of electrons. So they normally embed the redox cofactor quite deep within the protein structure, and that makes it more difficult to build electrical connection between the electrode and the protein. The other aspect about redox enzymes is that the catalytic activity strongly depends on the specific geometry of the amino acid that surround the cofactor, and so the three-dimensional structure of the protein is extremely important for the catalytic function of the enzyme. The second aspect is that you have dynamic effects. It's not a static system. There is dynamics also to be considered in the catalytic reactions. So these two aspects of dynamics of the protein structure and the protein structure itself that needs to be maintained in the native conditions is an important aspect, because when you immobilize the protein, you want to make sure that it preserves as much as possible the native structure and also the native ability to do this dynamic process, because otherwise you will affect the catalytic reaction. You can do that with different approaches. Two of these approaches are electrolysis engineering and protein engineering. In terms of applications of protein film electrochemistry, now in the literature you can find many applications. One is the use of protein field voltammetry for mechanistic studies, so to study the properties of the enzyme. This is what Max has shown for the hydrogenases. You can use these techniques to construct biosensors, in which case you have to make sure that the turnover of specific analyte is proportional to the current that you are measuring. There are now examples of this system used for constructing renewable energy systems. Some of these systems we can debate on their validity. Lastly, there is the use of this technique for electrosynthesis. That means interfacing enzyme with electrodes to drive the specific production of products of interest. Now, as Maxi already touched upon this, you can have either a direct tethering of the proteins on the electrode surface, and so you can drive directly the electron transfer from the electrode to the cofactors. So this can be difficult, especially when these cofactors are deeply buried within the protein matrix. Then in this case, sometimes you are forced to use redox mediators. So a molecule that are free in solution and they are shutting in electrons between the electrode and the protein. This complicates extremely the pictures. It's much more difficult to get mechanistic information, and in terms of applications, it's also increasing the cost of whatever you are trying to do. Now, if we're looking at the examples of what we can use for protein film electrochemistry, and so we can either study redox proteins or redox enzyme. Redox, in the case of redox proteins, normally are small proteins that are involved in electron transfer steps along, for example, the respiratory chain. They are normally quite robust in terms of the scaffold of the proteins. They are small, as I said. Quite often, the redox cofactor is partially exposed to the surface, or if in any case, it's very close to the surface of the protein. So they are quite relatively easy to study because you can immobilize them being quite robust without affecting the structure, and because the cofactor is exposed to the surface, it's actually also quite easy to build the electrical connection between the electrode and the protein. Much more interesting, obviously, is to try to study redox enzyme. In this case, as we said before, the structure, the dynamics is important to maintain the catalytic reaction. The cofactor is very often embedded deeply inside the protein because the protein tries to avoid spurious reactions, so the catalytic site and the cofactor are embedded within the structure away from any possible interaction with molecule within the cell. Quite often, there are multi-subunits, so they're quite big, and so it's quite difficult. It's much more difficult if you want to tether them to electrosurfaces and get information from these enzymes. But it's more difficult, but not impossible. And I will show you some example of this. In terms of the engineering of the electrode, you can choose many different approaches for mobilizing your proteins on the surface. You can directly mobilize the protein either, for example, on metal surfaces or on carbon surfaces. Also metal oxides, as Maxi already shown. You can include your protein into a layer of polymers that can be also a conductive polymer that helps you the conductivity between the electrical connection between the electrode and the immobilized protein. And you can also use self-assembly monolayers of different nature that you can construct. Normally on metal oxide, gold is the most studied. But most recently, you can also, there are examples also of self-assembly monolayer constructed on metal oxides as well as on carbon. And on top of the self-assembly monolayer, you can then choose to bind your protein either in a random orientation or in a oriented way to try to improve the electrical connectivity between the protein and the electrodes. There are in the literature examples of nanostructured materials used to try to improve the connectivity between electrode and the protein. One example for it is the use of metal nanoparticles, in which the idea is that this metal nanoparticle is connected very close to the surface of the protein, and so in this case, much closer to the reaction to the cofactor that is of interest for connecting the electron flow to the catalytic reaction, and then you tether this nanoparticle to the electrode surface, building the electrical connectivity between the electron and the protein. Other examples used nanotubes in different configurations. And then finally, you have the use of mesoporous materials that allows you not only to increase the load of protein bound on the surface, but also to have, if you want, a rougher surface that allows to increase the probability of productive interaction between the surface and the surface of the surface of the electrodes and the surface of the protein. I will not touch so much on this example of nanostructure. Mainly, I will give some example of the mesoporous material towards the end, but I will mainly focus on use of self-assembled monolayer and metal surfaces. Now, first, as an example of direct immobilization of metal electrodes, I have this example from the mobilization of a small cytochrome. So it's a small, as I said, robust protein. It's a cytochrome C553 from the sulfovibrio vulgaris. The heme is covalently bound to the protein structure, to the polypeptide chain. And interestingly, in this protein, it is partially exposed to the surface in two areas. By genetic engineering, we engineer specific cysteines located at either of these two portions of exposed heme. And then we mobilize the protein directly on gold, where it makes a covalent bond via the cysteine, because the sulfur makes a bond with 3 gold atoms. That is a covalent bond. And so we have the protein that is directly bound on the gold surface, is oriented, and it has this two different orientation, and then exposing, let's say, the portion of the heme that is exposed towards the electrode surface. We monitor the position of the protein by AFM. So here you can see here the gold surface before immobilization. You can see the triangular shapes here of the terraces 111 gold that's been annealed. Then you can then follow the position of the protein, and you can start seeing the formation of a monolayer, and then the more you leave it, the more aggregations you see on the surface, and the more proteins, let's say, build multi-layers on the surface. We were interested in exploring the monolayer, but we also observed that the multi-layer, they seem to have an interesting phenomena of inter-heme electron transfer along the multi-layer, but it was make it quite difficult to understand the behavior. So then, looking at the behavior of this protein in an electrode, we can see that they both shows a CV, so the shapes are different. The current is proportional to the scan rate, as Maxi said before, showing that both system, the protein is immobilized on the surface. The differences in terms of shape of the CV could probably be associated with differences in the behavior, practically on the capacitance current contribution, that is associated possibly with the different behavior of the proteins in the two different orientation on the electrode surface in terms of their response to the applied electric field, in terms of compressibility and behavior of the entire protein matrix that changes practically the contribution of the capacitive current. Now, instead, to give you a more detailed example of immobilizing an enzyme on an electrode surface, I'm going to talk about cytochrome P450s. So first, to give you a quick overview of what cytochrome P450s are. They are heme-containing proteins. They are called cytochromes, but they are not really cytochromes because they are enzymes. They are actually oxidases. And they use oxygen as a substrate to oxidize aliphatic chain and carbons that are non-activated, so quite difficult reactions. They are involved in the metabolism of all the drugs that we take, and actually they are responsible for the metabolism of every xenobiotics that is ingested into the body. And because of that, they are practically the testing of P450 activity is a prerogative for approval of any drug. So it's the interest of the pharmaceutical industry to develop assays for testing the new drugs for P450 activity. Though it's quite difficult to develop high throughput screening system based on cytochrome P450. So people have tried to develop different methods based on fluorescence, but it's not that simple. So to have an electrochemically driven platform for testing drugs before they go to animal testing would be an ideal scenario. And so that's why we were involved in to try to develop this biosensor. The cytochrome P450, as I said, is a heme binding protein and receive electrons from a redox partner that is the cytochrome P450 and ADPH dependent reductase. This protein has two cofactors, that is FMN and FAD, and take electron from ADPH. So practically these two cofactors convert a 2-electron donor that is an ADPH in 1 electron acceptor that is the heme. So it delivers 1 electron at a time. It's in a timely fashion to the enzyme, and we'll see after what it means. In eukaryotes, so in humans, these cytochrome P450 are membrane bound, and they are tethered to the membrane via a helix loop helix system. So if you want to study, you have to engineer them and cut this membrane out and the membrane anchor out practically. In terms of the catalytic activity, catalytic cycle, as you said, there is a heme in the active site. It's ion 3 normally, and the resting state. It collects 1 electron and goes to ion 2. Ion 2 has a higher affinity for oxygen, binds oxygen, and then a second electron comes in, and then 2 protons to form the ion 4 oxoform, that is compound 1, that is extremely reactive species that will then react with the aliphatic chain and oxidizing it. And then the protein goes back to this resting state. Now, as you can see from here, there are several routes for these, let's say, for these intermediates to be lost. And if electrons and protons don't come in at specific timing along the catalytic cycle, there is the risk of losing these intermediates. And the loss of these intermediates leads to the formation of reactive oxygen species. So if you lose the intermediate after the first electron, you will generate superoxide. If you lose the intermediate after the second electron, you generate hydrogen peroxide. And this could be dangerous for the cell and for the system because it creates damage. So what we did, we mobilized cytochrome P450, just the catalytic unit, just the heme binding protein on an electrosurface. Initially, we used a polymer to create a film on the carbon surface. And in here, we can see CVs of non-catalytic peaks. And when we add a specific substrate for the enzyme, we can see that we get a catalytic effect. And we can also measure the formation of a product, and the product that is formed is the correct one. And when we look at the CVs, we can see that there are 2 peaks, one at minus 85 millivolts and one at plus 60 millivolts. And this is indicative that there is heterogeneity within the film. So the protein is experiencing different environments, and so that it gives you 2 different reduction potential because of the different environment. So ideally, you don't want to be in this situation. You want to have the protein in one specific form. So what we did is to look at the structure of the protein, and we saw that there are specifically exposed cysteines, uniquely exposed cysteine, only on one side. So these are the 2 faces of the protein. This is the substrate binding site, and this is where the reductase will normally bind. And on the face where the reductase will bind, there are 2 exposed cysteines that we can use to anchor the protein to the electrode surface. So directed immobilization on gold doesn't work, but what we can do is that you can make a surface monomer layer on gold and then tether the protein to the surface monomer layer. So we use the two-stage surface monomer layer formation, initially with cysteamine, and then the amine group of the cysteamine react with this cysteamine malamide group. Then the succinimide malamide group. Then this reacts with it, and the succinimide group is leaving a malamide group exposed to the surface of the surface monomer layer, and this malamide group specifically bind the cysteine. And so then you can have covalent binding of your protein to the electrode surface via the surface monomer layer. And in this case, we get a much better CV with only one peak, and when you subtract the capacitance current, you see that they are quite nicely overlap peaks. We can then titrate a substrate and measure catalytic current, and from the catalytic current, plot the catalytic current in function of the concentration of substrate, and get a Michaelis-Menten behavior that gives us a Km that is very close to the Km in solution. And also we can then measure the product formation, and again is the correct products. So what you can do practically is that you have your protein immobilized on the electrode surface via the surface monomer layer, and then you progressively add substrates, and then you get the catalytic current progressively increase with the increasing concentration of substrate, and then you can reconstruct your Michaelis-Menten curve. And what we can see when we compare all these different immobilization methods, we see that the one that gives the best performance in terms of product formation is when we have the protein covalently bound and oriented on the electrode surface. We then did genetic engineering to try to take away specifically one of the two cysteines, so to leave only one anchoring point. And when there is only one anchoring point on the protein, then the performance in terms of product formed increases further. So practically, again, reducing heterogeneity by taking away the two anchoring points and leaving only one improves the performance of the system. When this is still not enough, because the tethering enzymes is more complicated, then we can think about in doing protein engineering to try to improve this. To give you an example of this is nitrogenase. People have immobilized the catalytic unit of the nitrogenase on the electrode surface, and in this case they can electrically connect this catalytic unit to the electrode, and they can see very beautiful electrochemical performance. They can see the non-catalytic peaks in the CVs of the different cofactors in this unit. The problem is that when they try to do catalysis with it, they try to drive the catalysis, then they lose the ability to distinguish between hydrogen and nitrogen. So one of the strengths of the nitrogenase is the fact that it is capable of distinguishing between hydrogen and nitrogen, and so it can specifically reduce, it can specifically catalyze the formation of ammonia from nitrogen. This ability to distinguish between these two substrates was lost when this unit is directly connected to the electrode. When they instead interface on the electrode surface, the catalytic unit together with the natural electron transfer partner of the nitrogenase, they were able to have correct catalysis and correct distinguishing between hydrogen and nitrogen. Nevertheless, because this now is a much bigger complex and the cofactor into the electron transfer partner are buried, it is not possible to have direct electron transfer from the electrode to the protein. And so they were forced to use mediator to be able to connect this massive system. So you can see here the importance of the redox partners in delivering electrons, because as we say, in terms of the catalysis, it's not just the simple event of delivering the electrons. They have to be delivered at a specific time and in specific manner. And then for catalysis, you can also have the importance of dynamics, of conformational changes. And these can be driven or facilitated by the protein partner. So you need to think about that if you want to try to construct the whole system on the electrode, you have to take this into account. And so I'm going to give you an example now of how protein engineering can improve. So we looked at the cytochrome P450 and the redox partner, that is this protein that has these two cofactors, this reductase. So what we did, we took the catalytic unit of the cytochrome P450, and then we connect to this catalytic unit either a simplified version of the reductase, that is a flavin-containing protein, so practically a protein that has only one of the two cofactors that are switching from a two-electrode system to a one-electrode system. So it's practically still containing a relay, but it's only one relay. And another example in which we bind to the catalytic unit the entire reductase. Okay, so the entire unit. So this redox partner is bound to the catalytic unit. And in terms of initial proof of concept, we see that when we bind an electron transfer partner to the catalytic unit, we can see that we get an increase in the product formation. So we can see that there is an improvement of the catalytic performance. And then when we put everything together, we look at the effect of different immobilizations, so different electrode structures, so different electrode surfaces, and orientations versus random immobilization, together with the engineering of the proteins to attach the electron transfer partner to the catalytic unit. We see that engineering of the electrode to have direct and oriented immobilization helps. And on top of that, also we have an improvement with engineering of the protein. So by combining engineering of the electrode and engineering of the protein, you get the highest, let's say, level of catalytic performance of the system. And then when we look at the performance of the system, we see that the systems that has the electron transfer partner fused together with the catalytic unit produces less reactive oxygen species. So the reason why you get an improved performance in catalytic performance is because the electrons are delivered in a correct way in terms of the timing, so that it leads to less waste of electrons into generating non-specific products, in this particular case reactive oxygen species, but instead the electrons are driven and used productively for the formation of the product. So practically by combining this engineering aspect, the engineering of the electron, the engineering of the protein, you can drastically improve the catalytic performance of your immobilized protein. So this is just a quick conclusion of the first part. And then we can move on to the second part. That is instead to try to understand the variables in the protein immobilization and how we can use or tackle this variable to try to improve the mobilization of the protein on electrode surface. So I give here a few examples, possible variables that you can tackle. Surface cleanliness, that is what Maxi also mentioned before. You have the surface morphology that can affect the behavior of the protein on the surface. And then there are some specific properties of the surface mineral layer that we can look at a little bit more in detail. So in terms of the surface cleanliness, we can see that when we look at oxygen plasma cleaned gold surfaces with AFM, you can see that they are nicely and smooth. But if we leave them outside for 7 hours, for example, exposed to air, immediately you get the absorption of a lot of particles and organics on the surface. And we can see that also by doing a FTIR measurement, and we can see the oxides formation as well as different organics depositing. The point is that the presence of these organics and particles on the surface will affect the formation of the cephaloseminal layer as well as the formation of the protein layer, creating defects on the surface. And these have deleterious effect on the performance of the immobilized protein on electron. You can see here that the untreated, let's say the dirty electrode, responds much less to the presence of the substrate, and practically the sensor is not really working because the catalytic performance is strongly affected negatively. So it's quite important to clean the surfaces properly before you start forming the cephaloseminal layer as well as forming the protein layer. For gold, there are different methods. The best that we see is that we tested is oxygen plasma. It's the most reproducible, the more gentle on the surface. Then moving to the surface morphology. If we think about gold surfaces, especially if we think about gold surfaces that we can use in an industrial scale to try to produce a sensor that we can then sell for a few pennies and not hundreds of dollars each, then in this case there are several surfaces of gold that we need to consider. And these surfaces, this gold material, will be very different in their properties. So you can have, for example, evaporated gold on silicon oxide. You can have evaporated gold just on silicon. You can have gold on glass that is being annealed. You can have screen printed gold. You can have spattered gold on plastic. Spattered gold on silicon oxide. You can have spattered gold on plastic. And all of these, they have very different morphology and very different properties. And you have to consider which one will be best for you and which one will affect the performance of your immobilized protein. So in terms of examples of what we did to test that, we looked at two different surfaces. One is gold evaporated at 300 degrees on silicon oxide that is partially annealed while is immobilizing. And so it forms this kind of very big grain structures. And then you have gold evaporated at room temperature on native silica that instead it forms these kind of very small particles of gold on the surface. And when we look at them, you see that the structure is very different between the two gold structures. One has very small grains on the surface. This is very big grains on this one. And when we immobilize the cephaloseminal layer and the protein on the surface, and then we test the performance of the enzyme on the protein on the surface, we see that suddenly one of them doesn't respond anymore, while instead the other one is still responding to the presence of the substrate, and so it still gives you a catalytic current. So the question is why the two surfaces are so different in behaving in terms of forming immobilized protein that respond correctly to the system, to the substrate. So to do that, we try to look at the formation of the protein layer onto the surface. Now it's complicated to look at the formation of the protein layer directly on these very rough surfaces because they already are rough. So you already have these particles on the surface, and then you're trying to see the protein immobilizing. You can see obviously that the protein is immobilizing on the surface, and that if you leave it for longer, it starts to fill in these large grooves into the surface. But it's very difficult then to try to look at the formation of the protein layer on top of something that is already rough. And so you can see here a section, not a section, but the profile, of the AFM. You can see that there are the proteins on top of something that could well represent the gold surfaces, but it's very difficult, obviously, to make something out of it, or to make any statistical analysis. So we went back to look to work on gold that is atomically flat to start with, and then look at the formation of the protein layer on that. And instead of using the classical annealed gold on glass, we use this method that practically evaporates gold on mica, and then you strip away the mica, and where the mica was, let's say, touching the gold, is a completely atomically flat surface. And you can see here, this is the surface of the gold. It maintains the grain structure, but each grain is atomically flat. And then on this, we can then study the formation of the self-assembly layer on the surface, and then the formation of the protein layer, and then follow that with different concentration of protein, different times, and then you can look at the formation of the monolayer of protein and the multilayer. Together with that, we can also use the phase that is obtained together when you're working in tapping mode in AFM. Because this gives you an indication of the different type of interaction of the tip with the surface. And this interaction changes when the tip is interacting with a soft material like the protein, compared to, for example, the self-assembly layer and the gold, bare gold itself. So you can guide yourself into try to, let's say, assign the particles that you see from the tapping mode measurement, and then together with the phase to try to see these particles, yes, they could be protein because the phase changed and the tip was interacting differentially with that. So by doing that, we then did a study of the formation of the layer on the surface, and we can see that when we look at the self-assembly layer, we get a Gaussian distribution with centered with a height about 0.6 nanometer that correspond to the height of the self-assembly layer. And then when we add the protein, we can see that this Gaussian distribution shifts to higher value, though initially it's not completely shifted. So you still have some height corresponding to the self-assembly layer and some higher height corresponding to the protein. So obviously you start forming the layer, but it's not a uniform layer. There is still patches of self-assembly layer exposed. And then when you leave the protein for longer, or you increase the concentration of the protein, you get multiple distributions of the height that suggests that you start forming the layer protein, but you also form aggregates on the surface. So there are sites in which you start forming aggregation before you start forming a full uniform layer. So to try to put into context of what could control the formation of these aggregates and what practically controls the mobilization of the protein, and why these different surfaces give such a difference in terms of performance of the protein, of the sensor. Then we can look at the section here, for example, at the profile of the gold surfaces, and you can see that the gold surfaces with a large particle is very different than the one with a smaller particle, and this gives you an indication of the size of the protein with respect to the grains of gold. Now, normally people, when they look at surfaces, they tend to use roughness as an indicator of the quality of the surface. And the problem with roughness is that it only takes into account the height changes, the difference between the peak and the trough. And so, for example, these three surfaces here, they all have the same roughness, but they have a very different frequency of grain boundaries. So what we are interested in instead is to try to address the number of grain boundaries into these different gold surfaces. Between these surfaces here, you can see that the number of grain boundaries is much smaller in the one with a bigger particle compared to the one with a smaller particle. So what we did to try to study the behavior of the mobilization of the protein on our platform that is using the the flat gold, yes, atomically flat gold, but still maintains the grain structure. Then to try to address that, we use subsection of the images and then use cross sections and then did a statistical analysis and saw that the distribution of the grain boundaries matches the distribution of the protein particles. So the protein initially binds at the grain boundaries, and these becomes also sites of aggregation. So if you have a lot of grain boundaries, the result, you're going to have a lot of aggregation points, and then the resulting layer of protein is not going to perform well. And that's why the difference here, the one with a high number of grain boundaries didn't perform well at all. The one with a low number of grain boundaries instead performed well. So there is this kind of correlation. Now, in terms of the characterization of the self-assembled monolayer, just checking the time, we can then study the formation of the self-assembled monolayer. Apart from using atomic force microscope, we can use FTIR, and we can look at the formation of the self-assembled monolayer and the layer of protein on top. And in this case, we can use grazing angle attenuated total reflectance FTIR to study the amide 1 band of the protein immobilized on the surface. And what we can see is that depending on the type of immobilization, the convolution of the amide band shows changes in the distribution of the component of the different bands that form the amide 1 band. And what we can see is that when we look at the direct immobilization on gold, we see that the distribution of these components is different between the protein in solution and the protein immobilized on gold. And the protein immobilized on gold is much closer in terms of the distribution of these bands to the protein that is being denatured with guanidinium hydrochloride. The suggestion then means that the protein, if you immobilize the protein directly on gold, the protein denatured. And that's what I was saying, that you need to be careful about maintaining the native structure. While instead, if you immobilize the protein onto the self-assembled monolayer, you can see that the distribution of the band is the same in solution and on top of the self-assembled monolayer, suggesting that the protein on the self-assembled monolayer instead maintains the native structure. So you build the correct interface between the electrode and the protein. And you can also use FTIR to look at the formation of the self-assembled monolayer. In this case, looking at the bands corresponding to the different chemical components of your self-assembled monolayer. And in this case, you can see that you get the amine band corresponding to the cysteamine, and you get the peak corresponding to the malimido group here. And then when you look at the entire self-assembled layer, you can see that there is a combination of the two peaks, suggesting that you don't bind the malimido group to every single cysteamine on the surface. You get steric hindrance, so there will be practically a carpet of cysteamine underneath, and then some malimido group bound in different position. So you get a mixed surface on the surface. And you can see that also with AFM, and if you do STM, you can see that more clearly. And then you can also use this system, again, combining AFM with FTIR to try to see when you start forming, for example, aggregates, because you don't control the formation of the self-assembled monolayer, then all of a sudden, again, you start to lead to the denaturation of the protein. So again, you can see that aggregates or defects on the formation of the self-assembled monolayer lead to denaturation of the protein, leads to loss of activity, and then you practically don't have a working system anymore. The other thing you can do is that obviously you can use the self-assembled monolayer to try to insulate your system. It's quite important, for example, for P450s, because they use oxygen as substrate, and you don't want to have the oxygen directly reduced on the electrode surface, because you need anyway to scan to quite negative potential. So we changed the length of the self-assembly layer. Instead of this systeming, we used longer aliphatic chains, and then changed practically the length of the system to make it more insulated. But obviously, then you have to be careful about the distance between the electrode and the protein to maintain good electrical connectivity and good electron transfer. So in conclusion, on the second part is that the grain boundaries controls the SAM defects formation and also controls where the protein binds and the potential aggregation sites. So you have to be careful about the morphology practically of your surface. You can use different spectroscopic techniques in combination with AFM to try to look at the formation of the self-assembly layer, as well as the formation of the protein layer. And just to say that in terms of the self-assembly layer, you have to remember that they are not as drawn, lie nicely like a carpet, all very nicely packed. But you have to be careful when you start forming the self-assembly layer, because if you don't control the chemistry, the chemical reaction, you can lead to formation of this disrupted structure. Like in this case, the author of this paper suggested that the head groups bend and start binding on the electrode surface, on the gold surface, as well as the cysteine part, or forms bridges between the different head groups. So practically you need to be careful when you start forming self-assembly layer to control the chemistry, to control the formation of the layer. And then finally, on the third part, I'm just checking the time. I probably need to go a bit faster because it's already the end. I'll give you a couple examples of looking at much larger systems. In this particular case, photosystem 2, that is yes an enzyme, but is an enzyme made of many subunits, so it's a very large protein, and to electrically connecting this large protein is much more complicated. So in photosystem 2 is the water plus the quinone photooxidoreductase is responsible for it's using the energy of the photon to drive charge separation. So it contains in a reaction center a series of cofactors and series that absorb light, and you can then drive charge separation. You can then drive charge separation, and the electrons move in one direction towards the quinone, and a positive charge moves in the other direction, and you have electron transfer from the tyrosine and then from the catalytic side, and then you have accumulation of 4 holes at the manganese cluster for driving the oxidation of water, and then you have on the other side a collection of electrons at the level of the quinone, and then after two electrons are collected, the quinone is then moved out into the membrane. So if you want to study this system electrochemically, you need to immobilize an electrode, maintaining the entire structure, but also you have to be able to electrically connect with the electrons route that is practically the quinones. You can, if you want, use a mediator, a molecule that will replace the mobile quinone and then shuttle electrons in and out of the electrode, in and out of the protein to the electrode, but you might want to avoid that. But then you can also test where the electrons are coming from, because then you can use herbicides that can block the side of the mobile quinone, and so then you can then distinguish if the electrons are coming from a mobile entity or directly or from the species that is directly connected on the electrode surface. In terms of the why you want to immobilize photosystem 2 on electrode, the idea is that people suggested that you can then construct systems, a bio-hybrid system that will allow you to source, let's say, different alternative energy sources, but is highly debated because the lifetime of the protein, once it's isolated from the cell, is very short. And because these reactions, especially the oxidation of water and the charge separation event, can lead to the formation of reactive oxygen species that damage the protein, then the lifetime of the protein is only half an hour. So in this case, it's quite difficult to make a system that is long-lived. But instead, we could think about having a system that is vectorialized. So one of the problem of the protein is that it normally lives in the membrane, and it has two phases, one where the electrons are moving and the other one where the catalysis of the water oxidation occurs. So you can imagine it's like two enzymes joined together, the two phases of the protein, of this enzyme. And so when you isolate the protein, then it's more difficult to study the individual reactions, because if you are, let's say, using an electron acceptor that takes electron from one side, the risk is that it goes to the other side and donates electron to the catalytic unit. So in this case, it's quite difficult to do these studies in solution. So one idea is that if you're mobilizing on the surface, you have a vectorialized system. You take electrons all in one direction, so you can study the other reaction, let's say, without problems of electrons leakage going around and short-circuiting the system. So there are different ways in which photosystem II has been immobilized on electrodes. It's been immobilized directly on electrode surface, either directly on the surface electrode, metal electrode, or subassembly. In this case, you can get non-catalytic CVs, but the system is catalytically inactive. So the proteins, the immobilization affected the structure of the protein, and the protein is inactive from a catalytic point of view. The most successful one has been to immobilize on mesoporous metal oxides. And there are several examples of this. And the other examples are using polymers, especially conductive polymers like osmium-based polymers, and they've also been successful. So once you interface your enzyme with electrode, you can then drive, if you have it correctly oriented with the quinones facing the electrode surface, you can then extract. You can use light to induce charge separation. Electrons are going to the quinone, and then you can extract these electrons and then measure a current that is a photocurrent that is proportional to the amount of electrons extracted and practically to the amount of event of charge separation. And on the other side, you have the catalytic unit doing the water oxidation. So you cannot measure CVs, practically non-equilibrium CVs, non-catalytic CVs of the reduction of the quinone, but you can only measure the catalytic event. The distance from the quinone to the surface is about 10 angstrom. So if you have direct interaction with electrode, it's a distance that is still okay for direct electron transfer. And so in this configuration, people have measured the direct photocurrent. When you add an inhibitor to the quinone, in some cases you get a reduction of the current, suggesting that part of this electron flow is coming from the mobile quinone. And then when you use a mediator, then you get an enhancement of the current. But this enhancement of the current often is due to the presence of multilayers, because when you have multilayers on a sink, in terms of direct electron transfer, only the layer that is in contact with electron will give you electron transfer. All the others will be silent. But if you add a mediator, then all of them become active. And so you get increased currents from the layers that are not connected to the protein. So depending on what you are trying to do, then you can use or not mediator, depends on that. I will skip this because I will just go very briefly into the work of the group of Erwin Reisner, where they developed a very beautiful highly porous indium tin oxide mesoporous structure. And it's this one. Practically, they deposit indium tin oxide nanoparticles together with polystyrene beads, and then they sinter the electrode, and that leads to the destruction of the polystyrene beads, leaving the cavity, and then in the rest of the structure around it, then you get the sintering of the nanoparticle to form the mesoporous structure of the tin oxide. So you practically get a porous structure that has both very large pores coming from the polystyrene bead and smaller pores coming from the mesoporous indium tin oxide structure within this structure here. Okay, so in general, depending on the size of the indium tin oxide particles you used, then you get you can change the size of these pores. And if you use the 10 nanometer particles or 15 nanometer particles, then in this case the pore size will be too small for the photosystem to go inside the pores. But if you use larger particle, then like 40 nanometer particles, so in this case you can get also some photosystem to entering the smaller pore. But then you get a lot of photosystem to also into the larger pores. And here they used fluorescence microscopy to look at the formation, particularly at the distribution of the photosystem 2 within the porous material. And depending on the size of the large pores, they get either, if you use pores that are too big, they get a lot of aggregates within these larger pores, and that leads to a very inhomogeneous system that is not very good in terms of performance. But if instead they use slightly smaller pores, and this one 750 nanometers, then instead they get a nice uniform distribution of the photosystem 2 within these pores. Each pore is still full of photosystem 2, so you're going to have some photosystem 2 directly connected to the surface, some that will not be connected. But if you only look at the direct electron transfer, then you're only going to look at the one that are practically decorating the surfaces of these cavities. They also changed the surface of the mesoporous material by using a surface monomer layer, in this case on the indium tin oxide. They used different ones. They used one that was negatively charged to try to orient the protein based on their dipole orientation, and then they used an NHS EDC system to covalently bind the protein on the surface. And in this case, they see an improvement in the photocurrent measured depending on the system used. So first orientation, then covalently bound, even further improvement. And also what they see is that they increase the longevity of the system in terms of current measured in function of time. But you can see it's still only 1 hour. But they increase the stability of the system by covalently binding the protein on the electrode surface. Now, one thing that I would like to mention here is that there is something you have to be careful about when you do self-assembly monolayer on metal oxides, because they have the tendency to change the conduction band position. That's because depending on the electron withdrawing or electron donating properties of the head group of the self-assembly monolayer, you get an effect onto the conduction band of your semiconductor material. So depending on what you're using, then you just need to be careful by building self-assembly monolayer on metal oxide. And then what they studied different performances of the electrode on the surface. They saw, for example, that there was they measured the current in function of the potential, and they measured they tried to estimate practically the redox potential of the species that donates electron, in this case the quinone on the surface into photosystem 2. And they see that the current disappears at a potential lower than minus 50 millivolts. suggesting that the redox potential will be closer to that value. Is still too high compared to the redox potential of the quinone in the protein measured by other means of spectral electrochemistry. In this case, that is minus 147 millivolts. It's possible that it's due to the interaction of the protein with the surface, but it's also possible that there are some limitations associated also with the conductivity of the ITO that is could change when it's in a mesoporous structure, because you get non-uniform distribution of the dopant, and then in this case the full conductivity kind of decreases. But what is observed as well is that there were both positive photocurrent and negative photocurrent, and these negative currents they correlated with a phenomena associated with the formation of chlorophyll anions that they generate reactive oxygen species. These phenomena does not occur in vivo because the lifetime of the excited chlorophyll is too short. But it happens instead on the electrode because there is sufficient connectivity between the electrode surface and the surface of the protein to be able to abstract an electron from this chlorophyll. So we then we work in collaboration with them using a ring disc electrode system to try to dissect the phenomenology of formation of these reactive oxygen species versus the reactive oxygen species formed via the natural physiological phenomena in photosystem 2. And so we were able to distinguish between the events that are non-physiological from the events that are physiological in terms of the formation of the reactive oxygen species. So it was, I think, a nice way to use this system to distinguish between what is physiologically relevant and what is not physiologically relevant in terms of the mobilized protein. So just to conclude in terms of the final part is that to immobilize and characterize large protein is obviously much more difficult. There is now the advantage of using this mesoporous material that allows you to increase the, let's say, a little bit like the eggs into a egg basket, like you can surround the protein with the material so that you can increase your chances of having good connectivity between the electrode surface and the protein. At the same time, the interaction between the metal oxide surfaces and proteins is quite mild. Normally you can get initial electrostatic interaction and then you can formation of a covalent bond on the surface. And so it's kind of mimicking more closely the polymer in a certain way, polymer protein interaction that you get between redox partners in vivo. So unfortunately so there's still more work that can be done, and more of these large complexes, especially nitrogenase, that is quite interesting, that can be studied. And that's it. And this is just acknowledgement of the people I work with. Well, I worked with in the past for the P450 work with Professor Gilardi, and when I worked for Nanobiodesign, there was a spin-off that tried to commercialize that part, Jim Barber, for introducing me to photosystem 2 and all the work with Bill Rutherford, and then the collaboration with Ewan Reisner. That's it. Thank you.
I have a quick silly question.
That's all right.
For the hierarchical electrode you showed, like the one like temperature with polystyrene, okay.
Mm-hmm.
Does it just stay like freestanding itself, or like how robust is this structure?
It's deposited onto another glass surface, so okay. It's not freestanding.
Okay.
So I think it's an FTO surface. I don't remember. It might be something else. I don't remember now. But anyway, it could be FTO. Mm-hmm. Then you deposit your layer of nanoparticle with the beads in, and then you sinter everything, and then you create this structure that is bound to the FTO. Yeah, I don't think you will be able to. I don't know if now they have I think that they also did it on different surfaces because for the rotating the disc ring electrode, I think the disc might be carbon, so maybe you can deposit it on different surfaces. Yeah, so it should be fine.